have Dr. Richard Crooks from UT Austin. He did his undergrad at University of Illinois Urbana-Champaign and his PhD at Austin as well, and a postdoc at MIT. And he is now a professor in the Department of Chemistry and Biochemistry. He's won a number of different awards, including the Pittsburgh Analytical Chemistry Award and the Charles and Riley Award in Electroanalytical Chemistry. His lab studies a number of different things, including understanding how catalysts and nanoparticles work, applications of bipolar electrodes for chem biosensing, as well as low-cost paper-based origami sensors. And I think today he will talk about using paper diagnosis for early detection of bioweapons.
Great, thanks.
Dr.
Yeah,
Crooks.
thanks, Amy. Okay, so I do have some handouts here that I'm going to pass around. And when the moment comes, you'll have to share them, because I don't have enough for everybody. Because they're from the two-dimensional slides. They're a little hard to figure out what's going on. Let's see. I think you guys got one of these. There's two different things here. There's one. Okay, so share a little bit. Okay. So anyway, thanks a lot for the invitation. This is great for me to come and see what you guys are doing. I'm inspired that you're here at 6 o'clock on Thursday night. And I need to go back and maybe try to get something like this going at UT, because it's really nice to get the faculty out of the room and just do something more interesting yourselves. So I'm going to tell you just a little bit at the end about chemical weapons. We're just really getting going on that. But let me say a little bit about how we got into this project, because someone asked me about this earlier. My group really is focused mainly on pretty fundamental chemistry, DFT calculations, nanocatalysts and things like that. But every 2 years, we all, 16 of us or whatever it is, pack up and go up to Taos and have a little retreat, And the cost for that I cover, but the price is you have to give a 30 or 40-minute talk about what you're going to do for the next couple of years. Not what you've done, which is much easier to talk about, but what your plans are. And one of the guys in the group, and I'll show you a picture of him, really got interested in these paper sensors. And he showed up at this meeting, and he actually had some results and some really good ideas. And I didn't know anything about it, so I take no credit whatsoever for any of this. We've been working on it for about 3 years altogether. I'm an expert in electrochemistry, but I suspect many of you know a lot more about low-cost, point-of-care type sensing than I do. So please feel free to interrupt, correct, ask questions, okay? Don't be bashful. Okay, first though, I need to tell you, some of you know where Texas is, okay, but not everybody. So here's Texas, and if you look at Texas on election night, it won't be brown, it'll be red, okay? And there'll be one little blue dot right here, and that's where I live in Austin. This is what most people think Texas looks like, okay? But in fact, it looks more like this. This is the downtown area. This is the lake that goes through, or the river. It's sort of a river, it's sort of a lake that goes through downtown. This is Barton Springs Pool. Somebody's smiling over here that grew up in Austin. He's probably been in this pool many times. And this is just right in the downtown area. And then this is the university here. It's a big urban university. I don't know, there may be 50,000 students or something like that there, but it's packed with people. We have our problems. This is our governor, believe it or not, and his name is Rick Perry. And I say one thing about him. He and I run on the same trail. We both do trail running, not together. But, and he doesn't run with Texas Rangers. He runs by himself. and he's a good runner, for a 65 or whatever year old guy, he's actually pretty good. But he does carry a gun with him when he runs, believe it or not. And he runs with his dog, and a couple of years ago, or a year ago, a coyote came after his dog on the trail. There's coyotes all over the place. He claims came after his dog. And he pulled out his gun and killed the coyote. And this was the subsequent press conference. He was bragging about this. So he's a complete nutcase. Okay, who knows what this woman is doing? Any ideas? Any guesses?
Getting antibodies.
What's that?
Getting antibodies.
Cosmetic.
Not antibodies. Good guess, though. Good guess. It's like a little bit related to that, but no. This was the, what's that?
Cosmetic.
No, that's a great guess, but that's also wrong. I think probably some of you may know that not that long ago, within the last 15 years or so, Revlon was, to test their eye cosmetics, they would cut the eyelids off rabbits and then test their cosmetics. That's over. There were protests about that. And they managed to do it for 50 or 60 years. No, this is the first pregnancy test. And during, in these days, a woman would come into her doctor and say, I think I might be pregnant. And the doctor would have her pee in a cup, and then the pee would be sent out to a lab like this, and they would inject that urine into the rabbit. And then 2 or 3 days later, sacrifice the rabbit and look at the rabbit's ovaries. And if they were enlarged, then the woman was pregnant. So this was the first pregnancy test, okay? The first medical pregnancy test. And this was in the, in the 50s, and it was this HCG, which is a hormone that caused the enlarged ovaries in the bunny rabbit, okay? So this is what, this is the new version of that. This is happens to be the clear blue pregnancy test, and one of the probably 2 most successful point of care devices. The other, of course, being the, well, you tell me, what's the other one? Diabetes, yeah, absolutely, diabetes. So this is a really interesting story, and this is the best part right here. 3 years from design, the first design meeting to commercial launch. I actually work with one of the guys that was on the team and the lead inventor of the Clear Blue pregnancy test. He's an English guy named Ian Richards. And so he's told me this story that a company called Unilever in the UK, he's British, hired a bunch of people, interdisciplinary people, biochemists, chemists, mechanical engineers, industrial designers, all right out of their PhD. So in England, that means 25 years old. And none of them were over 30, and there were about 10 or 12, he said, and they came into an empty room, probably something like this, and the guy from Unilever came in and gave them their charge, which was to come up with a better pregnancy test. A cheaper one that was easier to use, and one that doctors would believe, okay? And from that very first meeting till commercial launch was just 3 years. And in the first year, they sold 10 million of them. So if you think you have a successful sensor story, this is your competition. This is how it works. Probably you all know this already, but I'll tell you anyway. So it's a lateral flow assay, and the division of Unilever that this company became, this was a division of Unilever, but it eventually was so successful it became its own company, and it was called Unipath. And the reason it was called Unipath was why? One path, right? So there's several different sections on a pregnancy test. This is what's inside this plastic case. There's a sample pad here on which a drop is placed, and that would be, urine, usually first morning urine, midstream. You have to do this right. It is possible to screw it up. And then there's a label here, and in the first incarnation of these, the labels were colored polystyrene beads. Now they're gold nanoparticles, believe it or not. And they're attached to an antibody, and so here comes the urine down here. That's moving the labels. And then here there's a control line, and the control line has a promiscuous antibody on it that picks up any other antibody that comes down. So this line is just to tell you that the assay seems to be working correctly, that enough urine was applied, to move the sample and the antibodies downstream. That's all it's good for. And then this antibody is against the HCG, and only in the presence of the target will the label form here. And so confirmation of pregnancy requires both of these lines here. One just to show the control, just to show that the sensor worked, and then this line to show that HCG was present. There's a million ways to. This assay looks bulletproof. There's a million ways to screw it up. HCG in pregnant women is, goes from 0 to exponential in 6 weeks, and you can get way too much HCG, and that tricks the assay into thinking it gives a negative result. So you have to use it, within a few weeks of actually becoming pregnant, or it doesn't work right. But it's nevertheless, they sell 10 million of them a year. Some of the reasons it's successful is because it's pretty simple. It's just a strip of nitrocellulose on a piece of plastic backing. That's one reason. Another reason is it's a yes-no sensor, and that's all you ever need. It doesn't need to be quantitative because what? You're not 38% pregnant. You're either pregnant or you're not pregnant. Okay? So it's qualitative, which is really good. And nobody's going to die if it's wrong. Okay, so the FDA testing and all that stuff's not really that important because this sensor is for a community called the worried well. These are people who aren't sick, but they think they might have something. In this case, they think they might be pregnant, but they're not sick. Okay, and that's a real popular market that the drug companies like to go after, the worried well. Okay. Anyway, there's two nice technical things about this. One is that you can really they pick labels here that you can see with your eye, so that's really good. And then the other thing is it's a little more subtle. Is this line, everything sticks to nitrocellulose, so all you need to do is spot these antibodies on. You don't need to do any fancy immobilization. They stick really well to nitrocellulose. And there's a very thin line here, and that's a form of concentration. So when these antibodies are coming down here, the labels are concentrated so you can see them. If that line was much wider, you wouldn't be able to see them because the color would be washed out because there'd be less color per square centimeter, for example. Okay? Okay, well that's about where things stood till George Whitesides showed up and changed everything, as he's done many times in many different fields. And he published this paper just 7 years ago, and he showed that you could do basically the same thing that I just showed you, but in 2 dimensions. And the way he makes these were now not nitrocellulose, just cellulose paper, so filter paper. And he used the tools of photolithography to define hydrophilic and hydrophobic regions of the paper. And then, he puts a colorimetric marker, in this location and this location. Those are the hydrophilic locations. And then the sample's applied here, and it moves up the paper, but it's confined, to move in this channel by this photoresist here and here, which is hydrophobic. So it moves up the channel, and then it distributes itself between these 3 reservoirs. No pumps or anything required, of course. It's just capillary flow. And one of these, in this case, was for protein, colorimetric protein indicator, and the other one was a glucose indicator. And you could see, you could do something approaching semi-quantitation just by looking at the extent of this is different amounts of BSA, which was the protein used, and then different amounts of glucose. And so, the detection sensitivity isn't great. It's not really quantitative, but it was a really nice idea and got lots and lots of people thinking, including this guy Hong Lu in my group. So we started playing around with this, you read, it's like everything else. You read the research paper and you go, oh, this looks like a piece of cake, right? And then you start actually doing it and you realize there's just tons and tons of problems. And this was the list of problems that, we came up with. And, the device fabrication can be surprisingly complex. And it says here, especially for three-dimensional paper fluidic devices. And some of you may know, Whiteside's also made three-dimensional devices. And he did this using the same approach, but making many layers, and then stacking those layers together and holding them together with two-sided tape. And you can't imagine how difficult it is to work with two-sided tape with precision laser drilled holes, and you're trying to line up this, with your fingers, this two-dimensional tape, on paper, right? And so, very complex, very difficult to do, and I don't think anybody does that, actually. I think there were 2 or 3 papers in the Whiteside's lab, and that was it on that. But anyway, there's lots of other things here. The fluids move really slowly. For point of care testing, people say 5 minutes. You got to be done in 5 minutes, or else no one's going to be interested. And the problem is fluids moving through paper move very slowly, and the further the fluid has to move, the more slowly it moves. Okay, and I'll show you an example of that. Anyway, you've probably read all these yourselves already, but this is another really key one here. It's hard to do wash steps, and who's ever heard of an immunoassay without a wash step? Okay, and how are you going to do a wash step on paper? And are you going to have this person who's doing this, point of care test get out their bottle of buffer and do some kind of wash? It doesn't make sense. So lots and lots of problems. One way of solving all these problems is to add complexity. But for paper sensors, the whole point is to make them for $1. And if you have to add complexity, you might as well use, PDMS or some kind of plastic, or to make the device, since you're going to lose the advantage of cheapness. Okay. So here's Hong, and the idea he came up with in our first paper in this area was based on origami. Well, Hong's Chinese, obviously, and I guess when all the American kids were out at recess, kicking a soccer ball around or something, he was doing origami back in China. And when he saw this paper by Whiteside's where they were sticking together two-sided tape and paper, he said, we can do this just by paper folding. And so that was our first contribution to this field. And a lot of nice things about it. First, it makes multi-dimensional, three-dimensional paper fluidic devices manufacturable. And it's really the first example of being able to do that. And then, the other thing is you can unfold it and you can read different assay results on different levels of the paper. And so we've done a fair amount of that kind of stuff. And I'm not going to talk about it today, but I just wanted to mention it briefly here, because it was the first thing we did, and I think it's still pretty important. Okay, so let me talk about some of these issues and how we've addressed them in something approaching real assays. When I say real assays, by the way, I want to be clear that to really convince somebody that you can do, actually do an assay, you have to do hundreds of them and get CVs and show that they work over and over and over, and and that the reagents are robust, that are on the piece of paper, and there's not much of that done. Most people in this field are doing what we're doing, is they're making widgets one at a time and showing that in a laboratory, with a PhD working on it, you can do this test, okay? But what you need to do it a few hundred times and show that it works over and over. But anyway, so we address some of these problems here. And I'll show you a couple of the sensors we've made. So the first one is based on work from Kevin Plaxco's lab. Kevin is UC Santa Barbara. And he's actually an astrobiologist, but he's gotten out of that field and now is a sensor guy. And here's the idea that he came up with. This is a concept for electrochemical sensing. So this is a gold electrode. This is a strand of DNA, and it happens to be an aptamer. Aptamers are sequences of DNA that recognize specific analytes, okay? And the DNA is terminated in a redox active molecule, which is methylene blue. So methylene blue can undergo electron exchange with the surface. It can be reduced by 2 electrons and oxidized by 2 electrons. And so the idea here is that this particular aptamer happens to be specific for cocaine, and in the presence of cocaine, the aptamer wraps around the cocaine, and in the process of doing that, it brings the average location of the redox tag closer to the electrode surface. So here its average distance is further away, in the presence of cocaine it's closer. And then you do detection by electrochemistry, and this is a plot. You can think of this, it's not quite a cyclic voltammogram, which probably a lot of you are familiar with, but it's like a cyclic voltammogram. It's a plot of current versus potential. And in this case, this is an on sensor, so this is without the cocaine. The current is kind of low, and then in the presence of cocaine, the current goes up by about 50%. The peak current goes up by about 50%. This is incredibly robust. This works in everything but whole blood. And the only problem with it is there aren't very many good aptamers. There's like 5 that people believe in, okay? And there are people who claim to have aptamers for just about everything, but when you go to try to use them, they don't work very well. That's the problem. So if there were lots of good aptamers out there, this would be a huge deal. This is the way Kevin does his experiments. It's the way most electrochemists do their experiments. There's a beaker and there's some, big commercial electrodes here, and all of this is done on the surface of a gold electrode. So I called Kevin, and I met him in a meeting, and I called him up and I said, hey, we'd like to try to do this on a paper sensor, and can we rip off your science here and do that? He said, I'd be delighted. So that's what we did. One thing that we needed to be able to do this, though, one of my students went out to his lab, and when she came back, I wanted her to see this with her own eyes, and when she came back, she said, there's a problem, and the problem is we have to time this reaction, and of the binding of the aptamer to the target. And you remember from that pregnancy test, you put a drop of urine or whatever on nitrocellulose, and it just moves at its own rate. There's no way to time it, its movement. And so we had to develop a way of doing that in paper, and for that, we borrowed this idea of the slip chip from Rustam Ismagilov down at Caltech. And this is real simple. Okay, Rustam's version of this is with glass, and it's for clinical lab assays, doing maybe 100 assays simultaneously in very small volumes. And the function of a slip chip, or we call it a slip pad when we use it, is you have a drop here. You could imagine that there's a reaction going on in this drop. I should say the orange area here is some hydrophobic material like wax. The open areas would then be just regular paper, hydrophilic paper. So you can let a reaction proceed here, and then if you want to then deliver that reaction product over here, you just move these 2 pieces of paper with respect to each other, and that opens up a via down here, and so the fluid can move over. Okay, and when we first started this, I talked to Rustam, and I said, how difficult is this to do? He says, it's a real pain in the butt. He said, we put special fluorocarbons on the glass and all of this stuff. And, but I thought, well, it's wax, right? We use wax to define the hydrophobic regions, and what's wax? It's slippery, it's hydrophobic, might work. And it turns out that it did work pretty well. So let me show you how we put those 2 concepts together and to make a sensor. Okay, so this shows the first one, and some of you have this. You can, feel free to rip it open if you can get it open. Some of these are open and some of them aren't. You can actually see what it looks like. There's 2 parts. There's a slip layer, and I'm going to show you every detail of the fabrication here in just a second. There's a slip layer and a base layer. There are 3 screen-printed carbon electrodes on the base layer, and then I'm going to talk a little bit more about what's in the slip layer. But this is just cut-out paper here, and this is just paper without any wax on it. So the light gray here is always going to be wax. We print that with a thermal wax printer. The white areas will always be just paper that's been removed. Okay, so there's a hole in the paper, and then the yellow is where there's just paper, hydrophilic paper. Okay, so here's how it goes. We start out with just paper, okay? And what you're seeing on the left here, this is a whole sheet of paper in which, maybe a dozen or 2 dozen devices can be made simultaneously. And then this on the right is just a single device from this whole bunch of them here. So, first thing is, this is a $700 commercial wax printer, and you take a piece of paper and you just run it through the wax printer, and that gives you, this layer, okay, here. This is one of these, and you can see this printer. These printers are made for making printing glossy photographs, and that's why it's wax, because it's shiny, looks nice. And it prints the wax on top of the paper, right? So you need to do a second step, which is to heat up. You just put the paper on a hot plate for a second, and when you do that, the wax melts through the paper. You need to have the width of the paper here is about 180 microns, and that 180 microns is where the channels will be, okay? And then you screen print onto the top of this wax now, on the base layer, 3 electrodes, and that's done just with a Hobby Lobby kit here. It costs, I think, $100 or something like that. You can do a whole bunch of them at once, okay? And then we don't do this anymore, but we used to paint on a little silver chloride paint as a reference electrode, but it turns out that's just not necessary. It turns out the carbon, for those of you who know about electrochemistry, you always need a reference electrode in electrochemistry, and the carbon itself is actually a pretty good reference electrode. For 5 minutes, it's okay, and that's all you need. And then there's a working electrode here, and a counter electrode. And we have to, right now we deposit, this is the one step I don't like, we deposit a little bit of gold, just electrodeposit it on the working electrode here. This is the electrochemical cell we use for doing that. It's just because we need a way to immobilize that aptamer, and the easiest way to do it is just to buy a thiolated piece of DNA and put it on gold. I'm sure there's a better way of doing this, but this is lab stuff. This is not for sale. So that's what we're doing. You can ignore that. It's just showing reproducibility. When you electrodeposit gold, this is a real learning experience for me. That when you screen print carbon, it turns out only a tiny fraction of that carbon is electroactive. The carbon ink that's used is little flakes of graphite in a binder, and mostly it's binder, and so very little of the surface is actually conductive. So when you electrodeposit gold on this surface, you don't get a smooth coating of gold, you get little nodules wherever there happens to be a little electroactive region on the carbon. So that's interesting. It's fine. It turns out the surface area of this gold, these nodules is pretty high. It's actually twice as high as the geometric area of the carbon itself. So there's plenty of surface area, plenty of places to bind DNA. These are just cyclic voltammograms of several different devices. This is just the classical electrochemistry of gold, just to show you it's there, and it's very reproducible. We can do it reproducibly. Okay, then you get a biopsy punch, and punch out a hole here, and add the aptamer here. So the aptamer, this is part of the device, is this aptamer. And so you buy that from a DNA foundry. Okay, so that's the base layer. How about the slip layer? Okay, so you print the slip layer. This region is paper, remember, and then here's this this gray area is wax. Melt it so it melts through. And then with a razor blade, we've gotten very fancy now. We have a laser cutter that you can buy it in a hobby shop. They use them for engraving, like little league you know, trophies and stuff like that. It's a couple $100. And you actually cut out this section of the paper, okay? Okay, and then you put it together. Now it's ready to put together. So you put the slip layer over the base layer, and then you fold this flap over this way. And if you've got one of these, if you can get it open, you'll see what it may not be possible unless you've got a knife or scissors or something. You fold that layer over there, and then it's sealed up in the laminate just to protect it for future use. Okay, and that's what you've got. This is what you have. Okay. Okay, so here's how it works when you're ready to do an assay. So you pull down, you know, this slip layer. It just moves very easily. You know, the wax. You know, when wax is pretty easy to melt, and when you begin to pull, there's a little resistance at first, but you know, there's friction generated with that first little pull, and that melts a very thin layer of wax on the surface, and then it moves smoothly after that. Okay, so you pull down this slip layer, and now what you've done, let me show you what you're seeing here is that, remember there was that in the slip layer, there was paper up here. And so if you look underneath, that's that paper section now. And that paper section is now connecting together all three of those electrodes. And so that's this piece of 180 micron thick piece of paper is the electrochemical cell. Okay? So now you just add buffer here, and you run a background voltammogram. Now you slip this layer back up, okay, and now you put your sample on here. So the sample would be the target, you know, may contain the target that you're looking for, and it would be the target for the particular aptamer that you pre-immobilized on that electrode. So this is the whole point of the slip pad. It has two functions. One, it allows you to get that background voltammogram, which is very important because these paper devices, you can't assume everyone's going to be like every other one. If you can do the control and the assay on the same device, it's much better. And then the second thing is you need to time this step here after you add, you know, the incubation step after you add the target. You need to give it a couple of minutes to, you know, for the aptamer to bind the target. Okay? Okay, so there the sample's been added. Now you're going to pull the slip layer down. And again, now remember there's this piece of paper here, and that piece of paper is now connecting all three electrodes again. That's the electrochemical cell. And you're ready to take now the second voltammogram, which is, you know, the assay voltammogram. Okay? And so so that's how it works. Let me show you two assays that my student Josephine did. One of these was for DNA. So she gets this hairpin DNA here. This is an off sensor. Here's the methylene blue here. It's close to the electrode surface, so it's, you know, easily exchanging electrons. And then in the presence of the target, which, you know, can get a toehold here and then unzip this stem region of the DNA, then after that happens, the average distance of the methylene blue is further away from the electrode, and so the current goes down. This, by the way, is an exact replicate of an experiment that Plaxco had done using, you know, a normal electrochemical cell, and you know, we wanted to do that because we wanted to benchmark what we could do with paper against what he did in a beaker. So these are the results. This is without the target present. The black curve here, the black voltammogram, so that's this case, and you can see, you know, the current's pretty high. And then after you have the target, it goes down about 50%. And actually, it's exactly what Plaxco got. It's the same result on a much simpler device. This is a binding curve. The limit of detection here is 30 nanomolar. I don't know if anybody cares about detecting DNA at the 30 nanomolar level, but if they do, we can do it for $1. Okay. And then this plot just shows the signal suppression as a function, so that the distance between this curve and this curve as a function of you know, the match between the target and the stem loop. So this is a perfect match here. This is one mismatch, two mismatches, and then a complete mismatch over here. So you can even distinguish, a mismatch from, one mismatch from perfect complementarity versus, perfect uncomplementarity, I guess you'd say. Okay, and then just to show, some breadth in this assay, Josephine looked at a protein, thrombin here. There's a good aptamer for thrombin. And so, here in this case, the methylene blue is another off sensor. So the methylene blue is down here near the electrode surface exchanging electrons. And then you add thrombin. Thrombin, the aptamer undergoes a conformational change in the presence of thrombin that keeps the methylene blue further away from the electrode on average. And so you can play the same game. Here's the, without thrombin, and then in the presence of thrombin, there's a current suppression. And, binding curve, and, 16 nanomolar is the limit of detection, which is similar to what I just showed you. These numbers are exactly the same, within, error of what Plaxco was able to do. So we felt very good about these results. With a real, inexpensive, simple type sensor, we can get results that are on par with, what's done with a, research laboratory apparatus. Okay, so that's the first story. Now I'm going to tell you the second story, and this relates to the other device that some of you have. It's a little more complicated, but it's actually what we're mainly pursuing now. So it addresses some of these problems that I mentioned here. Okay, so we have a group meeting every week, with the people working on these paper fluidics, the students that are working on these. And, the problem is, as I mentioned earlier, you put water or buffer or urine or whatever on paper, it moves slowly. And the question was, how can we get it to move more quickly? And, we've been gnashing our teeth trying to think up a way of doing that. And it just happened that somebody else in my group who was working on a different project was helping somebody on this project and decided that day to come to this subgroup meeting. And once again, we were having this discussion about what we could do to speed up fluid flow in paper. And this guy, his name's Stephen Fosdick, he says, why don't you just get, the problem is the paper, why don't you just get rid of the paper? Just cut it out and use hollow channels. And so, okay, that's a great idea. So that's what we did. And almost everything we do now is with these hollow channels. So let me just show you briefly. It's super simple, okay? So what you're looking at here, this is this gray area again is wax, okay? And then there's a hole punched here and a hole punched here. And then this is a hollow channel. So this is just cut out paper here, okay? The white is just, that paper's been removed with a razor blade or a laser cutter. And then all the gray is still wax. And then down here, this is the bottom layer. This part down here we call a hemichannel. The first time we did this bottom part looked just like the top part. So it was wax, and all of this region here, this bottom part was wax. And then when you put this together, it's actually not three separate pieces of paper. It's one piece that's folded. So it's like origami. And when you put a drop of water here, what happens? Answer: nothing, because the channel on all four sides of the channel was wax. Okay, it's hydrophobic. And so what you have to do is on this bottom piece, you print the wax at a lower grayscale, not 100% wax, but like 50% or 40% wax. And then when you melt it, it only melts part way into the paper, okay? So there's a thin, 70 micron hydrophilic layer here, and just on the bottom, and that's sufficient for capillary, action to move the fluid through the hollow channel. As you'll see, it's a lot faster though than moving through a channel filled with cellulose fibers. Okay, and that's really what's shown here. This is that device that I just showed you, and there's been I see I left off the number here. This is, I think, a 20 microliter droplet placed at the inlet, and that 20 microliter droplet exerts a pressure of 1.2 millibar. Okay? And then what you're looking at here is the distance the fluid travels as a function of time in a hollow channel, and then a regular, cellulose fiber-filled channel, which is the usual thing. And it turns out it's about 7 times faster. Okay, and this is the actual flow rate. And I mentioned earlier that when you put a drop of water on paper, it flows kind of quickly at first, but pretty soon it really slows down, and you can see that whether it's a hollow or cellulose fiber. You can see as time goes on, the flow rate really begins to decrease severely. So this is this is just to illustrate this. This isn't a very exciting video, but let me play it here anyway. Yeah. Okay. So this is just a drop of food coloring here, a dye, and this is the hollow channel. You see that dye has already made it to the ends of the hollow channel very quickly. The channel going this way is a cellulose channel, and it's the same length. Everything's exactly the same, except the channel is filled with cellulose, and you can see it takes 7 times longer to fill up, to get to the end. There it is, getting there. So the hollow channel is really, it's the difference. It's the difference if your fluidic part of your assay takes 2 minutes. And with a hollow channel, that's 14 minutes. With a filled, cellulose-filled channel, 14 minutes is a killer for point of care. So anyway, that was a nice development. Okay, the other thing is, my group is basically electrochemists, and for quantitative sensing, you just can't beat electrochemistry. For quantitation, for simplicity of the measurement apparatus, reproducibility, all these things are just great for electrochemistry, which of course is why all glucose sensors, or I think almost all glucose sensors, are electrochemical. So we thought, what happens in these 180, what's the electrochemistry like in these 180 micron thick hollow channels? And remarkably, without going into a lot of detail here, it looks just like what happens in a beaker. These are textbook cyclic voltammograms of ferrocene methanol, and it works very well. So that was nice because it means that we can, in these channels, use electrochemistry as a detection method. Okay, so let me show you the last thing here. This involves timing reactions, dealing with this problem, trying to do an immunoassay without a wash step, trying to get the limit of detection down. Okay, and this is the idea that we came up with for doing detection in an immunoassay. It's like an ELISA assay, but there's no enzyme. And why don't we want to use them? Why not just use enzymes? Enzymes are somewhat fragile, most of them, although certainly horseradish peroxidase and glucose oxidase are pretty bulletproof. They're still organic, biological molecules. The other thing is they're really slow. And again, you're trying to keep the time of the assay down, and if you're going to sit there and wait for HRP to turn over its substrate, that takes time, and time is the enemy. So this is what we came up with to solve those problems. It's real simple. These are silver nanoparticles here. They're 20 nanometers. We use ones that are 20 nanometers in diameter. They're commercially available, and you can make them. And you add a little permanganate, okay? And the permanganate oxidizes the silver into silver ions. And then you apply a negative potential to the electrode, and that deposits the silver onto the electrode surface, okay? And then you scan the potential of the electrode positive, and you reoxidize all this silver. Now, why would you want to do all that? Okay? Well, you want to get, in electrochemistry, if you can get all the silver, or whatever material it is, on the electrode rather than out in solution, then you don't have to worry about waiting for diffusion to measure the silver. And if you have to wait for silver to come to the electrode surface to measure how much of it's there, the limit of detection that you'll measure is orders of magnitude higher than this approach, where all the silver is right on the electrode and you don't have to wait for mass transfer. Okay, you just oxidize it. It's all right where you want it, right on the electrode surface, and you oxidize it. So in this step, from here to here, you record a voltammogram. This is just a standard cyclic voltammogram. The potential is being scanned from negative values to positive values. That results in oxidation of silver. And then the area under this voltammogram, this is a plot of, you can see current as a function of voltage, but the voltage is related to time through the scan rate. So it's really a plot of current as a function of time. So the integrated area under here is coulombs. Those coulombs, I get 1 electron for every atom of silver in the nanoparticle. And the nanoparticles, each nanoparticle has about 250,000 atoms. Okay? So in principle, there's a 250,000x amplification factor from this technique, and it's practically instantaneous. You don't have to wait for anything to happen. So let me show you an experiment that we did using this idea. We use this real simple composite, we call it. It's just a magnetic bead that's functionalized with streptavidin. So no immunoassay right now. This is just an initial test. This is just streptavidin here. This is biotin, and the biotin is conjugated to the silver nanoparticle. And we mix these together and form this thing, we just call it the composite. Okay? So it's just basically it's a magnetic bead linked to a 20 nanometer silver nanoparticle. The magnetic bead is a couple of microns in diameter. And so here's what we're going to do. Here's that hollow channel. This is the idea. This is the hollow channel, and this is the working electrode. This is the electrode where we're going to make a measurement. And then behind that electrode is a magnet, a little tiny, 1 millimeter in diameter rare earth magnet. And then here's this composite here, the magnetic bead, and in our case it's just a biotin streptavidin interaction, but I'll show you in a few minutes. There'd be a target here, in a real immunoassay, and this would be the label for that target. And the magnetic bead is very important because now when the sample's introduced and it starts moving down this hollow channel, the magnetic beads will be attracted to this magnet. This is the second form of amplification. The first form is oxidizing those silver nanoparticles. The second form, we learned from the pregnancy test, is if you can localize all of your labels in the same place, you can get a significant amplification factor. So as the fluid goes down here, magnetic beads are attracted to the working electrode under the magnet, but only if they're conjugated. In other words, if the target isn't present, then the labels just flow on down the channel. This eliminates the need for a washing step. So this was another really nice aspect of this approach. Okay, well, now we have these magnetic beads with labels, silver labels on them, and then we have to somehow figure out how to introduce this permanganate to oxidize the silver. I'll show you how we do that in a sec. And then once the silver is oxidized, reduce it onto the electrode surface, and then we strip it off and record that voltammogram I showed you a minute ago. So this is the other device you have. This one I don't think is sealed up. And you ought to take a look at this because it's almost impossible to explain on a slide. It's too complicated. This came out of the brain of Karen Skyda in my group. I claim no credit for this at all. She thought this up, and it's really amazing. And it works brilliantly. So there's 4 layers, and they're numbered here and from top to bottom. This is, again, one of these origami things that folds up. The dark areas here are wax. The yellow is the hemichannel. That's that partially waxed channel that allows the fluid to flow. This is a cutout region here. That's cut by the laser cutter. This is a drop of dispensed blue dye that's dispensed and then dried, and I'll show you why that's important in just a second. And these are the electrodes, and these are just the to those 3 electrodes that I've already told you about. And then there's an inlet and an outlet here. Those are just cut out with a laser cutter. And then this is a slip layer, and the slip layer has an open channel here, and then just this little tab sticking in here that's pink. And that little pink tab has permanganate pre-dispensed and dried on it. And then this is just a line of yellow wax, and we call it the slip line, and it tells you how far out to pull that slip layer when the time is right. And then, this is what it looks like when when you fold it up and we press it together between a couple of plexiglass sheets, okay? You can see here, this is the inlet, this is where the samples apply. This is the magnet. You're looking at the top now where that magnet is, and then right under the magnet, you can't see it because it's underneath, but is the working electrode. Okay, up here are the electrodes. And then here's that the outlet where that blue dye is. Okay. This is what we read it out with. This is something else we learned when Josephine was in Plaxco's lab. He and his group had come up with this thing called a cheap stat. Okay, electrochemists use potentiostats, so Plaxco called this a cheap stat. You can build it for $100, and it's got a USB cable here, and it's very easy to use. Okay, so here's how it works. This is the part that's impossible to understand unless you've got one of these in front of you. All right. So the sample, in this case, it's going to be this preformed composite, okay, is added at the inlet. And it moves down this hollow channel. You're looking at a side view here. It moves down this hollow channel. And if the labels are attached to the nanoparticles, and only if that's the case, the magnetic beads will all be attracted to the magnet here, and they'll bring along with them the labels, but only in the presence of the target. Okay, and so this flow continues until the sink here is filled. Once the sink is filled, flow stops. Okay, and now it's just a static solution. When the sink fills up, this blue dye that was dispensed in the second layer is pushed up to the top, and now you can see that blue dye. It's right here. This is after the device has been used. You can see that blue dye. That tells you flow has stopped. And now it's time to pull out this slip layer, okay? And I'll show you how this works. Again, there's nothing very dramatic to see here. But let me let's see. The sample will be added here. This is the whole assay, okay? So the sample's being added. It's moving down this hollow channel, okay? This is the magnet. The working electrode is underneath the magnet. Okay, and in a second you'll see this spot turns blue. That means flow has now ceased. Okay, so all the labels are underneath the magnet. Then you just pull the slip layer up. Okay, and what that's doing is it's putting that little tab of permanganate right under the working electrode. The permanganate dissolves and oxidizes those silver nanoparticles. That's one of the key steps, right? And then you hold, for 12 seconds, you hold the working electrode at a negative potential to replate all the silver under the working electrode, and then just strip it off. The whole thing's done in less than 5 minutes. It usually takes 2 or 3 minutes, depends on what exactly you're doing, but usually 2 or 3 minutes. And these are the data that come out. These are we call them stripping voltammograms because we're stripping the silver off the electrode. And these are for different concentrations of labels. Okay, and you can see the range here is from about 100 picomolar. And if you look over at this inset, these are showing the 2 lowest concentrations. This one right here, you can barely see that little peak there, but it's definitely, that peak is plenty big enough to be integrated. Yields a 700 femtomolar limit of detection. So 700 femtomolar in a device that costs $1.22 to make one at a time in the lab. And this is the calibration curve here. As a function of the concentration of the labels. You can see if you go out to higher concentration of the labels, this thing flattens out at the top here. That's just because we haven't added enough permanganate to that tab. You have to know where you want to be in the calibration curve so you can add enough permanganate to the tab. But if you know what the assay is and you know approximately what concentration of the target you're looking for, you just dispense more or less of that permanganate. Okay. Anyway, these are some of the results, the benchmarks for this. I don't know, so the amplification factor is, pretty high, 10 to the 5th. It only takes a few minutes. And these are some of the things that we're working on, some of the things we're working on now. And I have just one slide on ricin that I'll show you. So now, we made that composite in a vial outside the device and then just added it to the inlet in the data I just showed you. But of course, what we want to do is take the magnetic microbeads and the silver nanoparticles, that are pre-conjugated to antibodies, and put those in the inlet and dry them so that they're just sitting there and they can be stored for some period of time. And then when the target's added, or the solution that may or may not contain the target is added, it resolvates those reagents, this reagent and this reagent. And in the presence of, in this case, the ricin A chain, it binds the target, binds the label to the magnetic bead. And then from here, it's just exactly what I showed you, okay? But you want to do this all ultimately on the device. So that's what we're doing now. It's a one-step immunoassay. There's no washing of any kind. You just add the target, and this sandwich forms, and no washing is necessary, which is a big deal. And these are just our first data. This is unoptimized. It's as I mentioned, a one-step immunocomplex formation. And this is the calibration curve. So we have a detection limit of ricin of 78 picomolar. I don't know. I have to give credit to my colleague Andy Ellington, who provided one of the antibodies for this, the ricin antibodies, and then we get the other antibody from this thing called the critical, it says certified reagent program. It's the critical reagents program. 78 picomolar, I don't know if that's a little or a lot. We can't get the guys in, the people, our masters in Washington, to tell us really, what kinds of limit of detection they're looking for ricin. They're interested in ricin because every week or so, somebody in Washington, the president or somebody in Congress, gets an envelope with a white powder in it. And the problem is that white powders always claim to be ricin, okay? But it's usually what the guys in Washington, our handlers, call spoofs. And that's baking soda, foot powder is very common, powdered sugar is very common. Okay, so it's not really ricin. But it's a concern that it would be ricin, because why? Because ricin is extracted from the castor bean. And the castor bean in Austin, Texas grows along the creeks, okay, everywhere. And you can go on the web and you can get a recipe, a real simple recipe that anybody with a high school education can follow to extract ricin from those castor beans. You get everything you need, an incredibly dangerous toxin, and a bunch of rednecks, okay? So that's a problem. That's why they're interested. And how much they're looking for, and it's just not at all clear. But 78 picomolar in a device that costs $1, that's a pretty good limit of detection. It's as good as any other immunoassay. It's probably never going to get much better than that. That's about the lowest limit. So anyway, let me finish up. I've already talked a little too long, I think. But it's really fun to make these. This is what everybody in my group wants to do now. Nobody wants to do DFT calculations, okay? Nobody wants to do that kind of stuff anymore. Everybody wants to do this project. And so it is a lot of fun. These are, there are lots of problems, and I've tried to share some of those with you. And here are some of the methodologies or fabrication, tool type things that we've done to begin addressing these. I've talked about all of these except this last one on chip separations, and we're working on that too, because for detection, everything I've shown you here is with buffer. We have successfully analyzed for HCG and a couple of other LH, a couple other reproductive hormones in urine, human urine. And but what is everybody interested in? Blood, okay? And blood is there's going to have to be some kind of separation, because those blood cells are a pain in the butt. And there's still lots of problems. We don't know much at all about nonspecific adsorption. We're learning a little bit about urine here, but blood and plasma we're just starting to fool around with, and all we know is it's a problem at this point. And then the reader, you talk to the guys in Washington again, you know, and you tell them you need a reader, and, you know, their enthusiasm, you know, goes down because the soldiers already are carrying around enough junk, okay? And these guys, you know, the stories you hear, you know, you give them something, even a cell phone or something, and, you know, they take it apart and try and play, you know, iTunes with it, right? And so, you know, you can't really give them a cheap stat. It's not going to work. You know, they're going to try and, you know, do something else with it, or it's going to get wet or something, you know. So, so the question is, how do you do quantitative analysis in electrochemistry without a reader? So that's a problem. So we're working on all that. Anyway, this is my group. This is a picture we took last month. The two main people that worked on what I told you today, Josephine did that aptamer sensor. And there's Josephine right there. And this is Karen, who's, you know, mined that other sensor came out of. And so that's it. That's all I have to say. I'm happy if you have questions to, you know, answer questions. Dead silence. Yeah.
I'm wondering, do you care about selectivity here?
Well, you know, these are, yeah, of course we do, absolutely. And, you know, so the types of things, we've just gotten this ricin assay working. And what we're doing now is we're, you know, mixing it with these spoofs, you know, and you know, some related proteins and seeing how selective it is. But it just depends on how good the antibodies are. It's just like any other, you know, at this point, our device is as selective as any other immunoassay. It's no better, no worse, because it just all relies on the antibodies. And the question is, you know, how good are the antibodies you can get against the ricin A chain or the B chain? And we're fortunate to work with Andy Ellington. Andy actually is the guy that invented aptamers. And so half his group does aptamers, and the other half does makes antibodies of, you know, various flavors. And so he's optimized one of those ricin antibodies for us, and it works pretty well. But, you know, we need to, you know, I haven't shown you anything really. I showed you a little bit about CVs in one case. 13% is our typical error bar. But I haven't talked at all about large numbers of devices, like hundreds of devices. We haven't done any of that kind of stuff. We haven't challenged these sensors with a lot of different interferences. I mean, it's been difficult enough just with urine, you know, all the junk that's in urine. You know, you wouldn't believe it, you know, what all is in urine, you know. So yeah, those are all great questions. But, you know, the problem is, you say to a graduate student, everybody here is going to be able to identify with this, how would you like to just go do the same thing 300 times, you know? You know it works, and now you just want to know what the error bar looks like, right? No one wants to do that. They all want to go publish a research paper, and you can't publish that kind of stuff. You know, no one's interested except the funding agencies, right, are interested. So that's a problem. So I'm putting together an army of undergraduates. That's my plan. There's one of them right there, okay? And here's the other one here, Francisco. And that's what these guys are going to do, okay? They're going to come into the lab, and they love it, right? Because what? They're undergraduates. They're just learning, right? And so you give them a challenge. Get 10% CVs, you know, do whatever it takes. And they love it so far. We'll see how long that love affair lasts. It's just hard to get people interested in doing the same thing. It's a great question. Yeah?
Do you consider doing any multiplexing via different half-cell potentials?
Yeah. Boy, what a great question. Yes, we are. You know, one of the things that we have some funding for is analyzing 4 female reproductive hormones, okay, simultaneously. So, you know, first morning urine, detect these 4 hormones. And I was mentioning to some folks I was talking to up here earlier, the reason people are interested in this is that these reproductive hormones are really at very different levels in women, in, you know, different women. and then throughout their life, during different life stages, the amounts are different. And even morning to evening, the amounts are different. And the same is true for men. Testosterone levels vary by a factor of 5 from, they're high in the morning and then they're lower in the afternoon and evening. So, these hormones are just all over the place. And there's some evidence now that if you can do longitudinal testing of these hormones, in other words, test, at the same time of day, and the same day of the week, every week, throughout your lifetime, then you can measure very small changes, and these are often indicative of cancers like ovarian cancer. They're small changes, though, early on in the disease, and they don't ever, the changes don't ever go out of the population error bars. So you have to know what the baseline levels were for the individual, okay? So that's the motivation. And so your question was, what about multiplexing? And in fact, what we do is exactly what you're suggesting. We use a different kind of metal label. Rather than silver, we use copper oxide. And so we can detect simultaneously silver and copper oxide. That's just starting. We're just fooling around with it right now. But the other thing, that's true multiplexing, but there's spatial multiplexing, where you just would have 4 different channels and you detect each, you just split the sample into 4 channels. And use silver for all 4 of them. So that's probably the easier approach, but who wants to do that? That's just more of what we've done, right? They want to do the sexy thing, which is the true multiplexing. And so, yeah, absolutely. Yeah, normally you want to look at more than 1 marker if you can, 2, 3 markers. Yeah, very good point. Yeah.
Yeah, I have a question about your lower limb detection. So in terms of like the Ryson study where I think it was like 7-8 picomolar.
Yeah.
Do you have any sense of what's sort of keeping you from pushing it lower?
The antibodies.
Okay.
Yeah. If you look at immunoassays, these types of detection limits that we're seeing are common even for ELISA, and it's just the quality, the binding strength of the antibodies are defining it, and so that's the best I think we're ever going to be able to do with immunoassays. But there's nothing that we're doing that you can't do in an ELISA here, in a microtiter plate. The difference is maybe you can do this, in a doctor's office, and maybe you can do it in 5 minutes, or maybe you can do it in the developing world, or maybe, another thing we got a little funding to work on is, you guys have legalized marijuana out here? Is there legalized marijuana? Kind of quasi-legalized marijuana? I don't know. I smelled some walking up here from the art station. I can tell you that. Anyway, the police are really worried about this. Legalization is coming, okay, for sure. Everybody agrees with that. And, what are the cops going to do? Right now they've got breathalyzers. You may or may not know that if you get busted for drunken driving enough times, at least in Texas, it probably takes 4 or 5 times, they'll put an ignition lock on your car that's hooked up to a breathalyzer, and you got to blow into this thing or else the car won't start, okay? And there's a company that has a lock on these things called Smart Start. They've made a lot of money. I consult for them, and I can vouch for the fact that the founders are all wealthy. And what are they interested in now? They're interested in a cheap dollar sort of THC detection system, because now people are going to be, driving under the influence, but it's going to be the influence of marijuana and not, under the influence of alcohol. So the cops have great tests for alcohol. They have breathalyzers and you blow into it. They don't have any tests for THC. And so they would like to have one for THC and for cocaine, and could you please multiplex those and do it for, $1 and have it be right 100% of the time, and, is what they say. So, So did I answer your question? What did you ask?
Yeah, I wanted to know about the lower limb detection.
Yeah.
Sort of like a background issue.
Yeah.
Sort of antibody month.
Yeah, no, it's just the antibodies, and it's the same thing for THC and stuff. It's if you can find the right antibodies or make an aptamer for it or something like that, you can get down to the levels that I showed you, but probably not any lower than that.
Have you done any optimization around sort of the bead loading, like doing higher concentrations of antibody on the surface and how to push the reaction rate?
We do all that. Yeah, we do serial optimization. There's better ways of doing it, but we do serial optimization. We, first optimize, the number of antibodies per bead, and then the number of beads, and and then the number of antibodies on the silver nanoparticles, and then the number of silver nanoparticles. And by doing this, you can move the linear range of the assay around. Low limits of detection are not necessarily good. Okay, we're working with one of the pharmaceutical companies, and I was so proud of the fact that we had gotten down to this very low limit of detection for the marker they were interested in, and they said, well, yeah, you're about 2 orders of magnitude low, and where they want to be, we're flat, so we need to push to higher limits of detection, right? So turns out not everything is limit of detection. And for these types of assays, cancer markers, forget it. This is never going to work. We're never going to. You can't do PCR on a piece of paper. Or maybe you can, but it's going to be so much screwing around. You might as well not. That's what I think. So this is for, moderate concentrations, things you want to do every day. The cops want a THC sensor that they can put in the back of their car and pull out when they need it. That kind of stuff is where this is going to find a use. It's not going to be ultra low limits of detection. That's still going to have to go to a clinical lab, I think, at least for now. Yeah?
Why not do oxidation with the electrode?
Another great question. That was my only contribution to this project, was that exact suggestion, which turns out to be not a good idea. Okay, here's the problem. This is not to scale, okay? Remember, the beads are 2 microns, the nanoparticles are 20 nanometers. This bead is a mile away from the electrode, okay? And the electrode doesn't even know it's there. In fact, it was on a slide on a bullet, but I didn't even mention it. This is a zero background technique, okay, unlike ELISA. This is a true zero background technique because, you don't see any silver nanoparticles, zero current, absolutely zero current if the target isn't there, okay? And so, you can't, if you try to do, we did this exactly what I suggested, okay? And they went and did it, and they said, it doesn't work. And by the way, that was really a dumb idea that you had. And we've solved the problem with the permanganate. So, yeah. You think it's going to work, but it's just too far away. We actually spent a couple of months trying to put a conductive shell on the magnetic beads. That would do it, okay? If you could get the bead within a nanometer or 2 of the electrode, and the shell was conductive, then you could oxidize all these silver nanoparticles. And I had them do that. I wasn't going to give up easily. And turns out it's real hard to put a conductive shell on, reproducibly on a polystyrene-coated iron oxide particle. So anyway, they came up with a solution that works great. And so that's fine with me. Yeah. That's a very good question, though. And absolutely, I have the same misconception that might work. That was my only contribution, and it was wrong. What else? Anything else? Everybody had enough pizza and paper fluidic devices? Okay. Well, thank you all for the invitation and for very patiently listening to my story.